fat body is functions as the principal phase of enzymatic mechanism. Besides these tissues, inside muscles are active for the secretion of healthy hormones, leading to energy production. Carbohydrates and proteins. The carbohydrates have been already been storage and structure conjugated by glycolytics and glycogen participate in various physiological processes. The storing of carbohydrates. Glycogen is stored in large amounts of carbohydrates for flight and other tissues after transformation into nutrition. Glycogen stored in moist phosphorus in the fat body of locust and microtubular and stored on hemolymph and to limited extent in the fat. The concentration of trehalose varies from literally in the fish versus not to more than 70 g in the lipid. Trehalose is the energy source in insects, giving up twice the amount of energy per osmotically active particles as does glucose, which is important adaptation of insects engaged in glycolysis. Structural carbohydrates are represented by the chitin, polymer consisting of 150 N-acetyl glucose amino residues. These are three polymorphic glycoproteins, and the mixed carbohydrates are represented by glycoproteins and are distributed in various tissues and secretions. The silk proteins and yolk proteins are the best examples of glycoproteins in insects. Some glycoproteins are attached to cell membrane. In catabolism, the glycogen phosphorylase is responsible for the primary cleavage of glycogen in Cysticerca glyconesis. Glycogen concentrate decreased by 64% within 10 seconds after the inception of cell, and the fat body is engaged 5% into 2 hours of life as a result of phosphorylase activity. The trehalose is broken by the enzyme trehalase. The enzyme is specific for the alpha-alpha flight muscles, etc. The chitin is degraded by the molting fluid containing the enzymes chitinase, degrading the chitin molecule to cell chitobiosunics and beta-N-acetyl D-glucose amylase, hydrolyzing terminal non-reducing N-acetyl glucose. Produce the glycolysis is a long reaction occurring in various tissues and catalyzed by specific enzymes hexolinase, phosphofructokinase, then triose phosphate isomerase, glycerol-aldehyde phosphate dehydrogenase, pyruvate kinase. Glucose 6-phosphate dehydrogenase and 6-phosphogluconate dehydrogenase. Gluconeogenesis. The synthesis of glucose from amino acids or glycerol takes place with enzymatic reactions. The enzymes involved in gluconeogenesis are pyruvate carboxylase, fructose diphosphatase, etc. The synthesis of glycogen from glucose is catalyzed by glycogen synthesis. In the early synthesis of trehalose takes place by trehalose phosphate synthesis and trehalose phosphatase. In lipid metabolism, the lipids comprise a large group of heterogeneous compounds such as fatty acids, fatty acid esters of glycerol, acyl glycerols, and sphingosine. Phosphoglyceroids, nonpolar alcohol waxes, terpenes, steroids, prostaglandins, and hybrid molecules such as lipoproteins and glycolipids. The total lipid content in large number of insects varies inter and intraspecifically. Storage lipids. The storage lipids are acyl glycerol, particularly triacyl glycerol, which is the major source of metabolic energy in insects. The distribution of triglycerols varies according to demand of the insect, developmental stage, and also varying between tissues. The major lipid storage tissue is the fat body, in which more than 50% of the wet weight and 75% of the dry weight is represented by triacylglycerol. Large quantity of triacylglycerol is also accumulated in the developmental oocytes and eggs. Then triacylglycerol may also be present in the hemolymph, although in most species the dominant hemolymph lipid is diacylglycerol, which is bound to the carrier protein lipofuscin. Transport of lipids in the form of diacylglycerol is a specialized feature of insect biochemistry. 90% of hemolymph diacylglycerol is present in the 1,2 isomer. The fatty acids, both esterified fatty acids and non-esterified fatty acids, accumulate in all tissues and constitute a major metabolic reserve. There are various types of fatty acids in the insects. These are saturated fatty acids such as mystic acid, palmitic acid, and stearic acid. The monounsaturated fatty acids such as myristoyleic acid, palmitic acid, and oleic acid, and the polyunsaturated fatty acids such as lignoic acid and linoleic acid. Then structural lipids. Phospholipids are the major structural lipids and are essential component of cell membranes and distributed throughout all tissues of the insect body. The phosphatidylcholine and phosphatidylethanolamine are the major phospholipids in insects, accounting for about 70% of the total phospholipid content. In Hymenoptera, Lepidoptera, and Orthoptera, the first type of phospholipid, Phylindiptera, the second type predominate. Then sterols. The insect tissues are incapable of synthesizing sterols and need a dietary requirement for sterols. They are important structural component of biological membranes. Then cuticular lipids. They form thin lipid layer on the outer integumentary surface as a part of epicuticle. Then lipogenesis. Fatty acid biosynthesis pathway is carried out by two enzyme systems, acetyl-CoA carboxylase and fatty acid synthetase. The first enzyme catalyzes carboxylation by acetyl-CoA to the 3C malonyl-CoA. The condensation of malonyl-CoA with acetyl-CoA is catalyzed by the fatty acid synthesis. The reaction is continued until the capacity for mitochondrial synthesis of fatty acids has been reported in hospitals for reversal of oxidation. The acylglycerol formation in insects takes place through the biosynthetic pathway described for another animal groups. The enzymes involved in acylglycerol formation in insects are the alpha-glycerophosphatase and diacylglycerol transferase acid, alpha-glycerophosphatase acid. Similarly, phosphohydrolase activity has also been found in fat body and clitomucil of Hyophora cercosphere. The phospholipid biosynthesis occurs through a 16 pathway involving phosphorylation and phosphorylation kinase. The phosphorylated base is then activated and transferred to 1,2-diacylglycerols to form the appropriate phosphoracylglycerol. The major site of the phosphoglycerol synthesis is the fat body, but may occur in the nerve tissue, flight muscle, and gut. Amino acids and protein metabolism. Hemolymph contain high concentration of amino acids and proteins. Amino acids. Insect hemolymph is characterized by high concentration of free amino acid, 100 to 300 times higher than that in the human blood. Significance might be related to repeated molting, where protein synthesis is cyclical, yolk synthesis and other active substances are involved in reproduction, osmoregulation, natural coordination, detoxification, phospholipid synthesis and energy production, etc. The composition of free amino acids in species specific in insects. Cysteine, hydroxyproline, methionine sulfoxide, ornithine and aminobutyrate are of much less common occurrence than other components. The free amino acid pattern in insect is tissue and sex specific. The glutamate content is 10 times higher in tissues than that in the hemolymph in Drosophila, while the adult female Culex contains 5 times more methionine sulfoxide than the male and beta alanine in reverse proportion. The amino acids are rapidly decreasing during starvation, while feeding elevates the amino acid concentration. In the developing eggs, qualitative and quantitative increase in amino acid content occurs at the initial phase and reaches at the maximum level at the advanced stage, mostly due to breakdown of yolk reserve to yield amino acids for the synthesis of tissue proteins. Total quantity of free amino acids increases rapidly after the oviposition. It varies greatly during the post-embryonic development. At metamorphosis, the total free amino acid exhibit initial rise followed by a fall during the latest stage of development, resulting in the breakdown of tissues of the preceding instars and formation of succeeding instar tissue. In the adult, both male and female, the concentration of free amino acids is increasing before copulation while decreasing thereafter as a result of their involvement in leukoprotein synthesis and accessory gland secretion. Aminotransferase activity occurs in the insect. Molecular weight of some of the aminotransferase enzymes has been determined. The most important physiologically significant one is the L-alanine aminotransferase, which maintains the amino acid pool at a proper level for protein synthesis as well as supplies metabolites for energy metabolism and catalyzes interaction between protein and carbohydrate metabolism. Tyrosine and beta-alanine are involved in the process of sclerotization and therefore occur in higher concentration before each molt during the post-embryonic development. Besides tyrosine, proline and tryptophan occur in large quantity in the insect hemolymph, which serve as a source of metabolic energy and homochrome precursor respectively. Proline participate in flight metabolism and provide intermediates of the citric acid cycle as acts as a primary energy substrate. Adipokinetic hormone increases the rate of protein synthesis in the fat body, suggesting hormonal regulation of protein synthesis in insects. Proteins are synthesized in the fat body and are released into hemolymph, and their concentration increases in the last larval instar. The larval proteins are often undetected in the adult stage. They are in two types: larval hemolymph protein or LHP. Hall's storage proteins are of an average range of 5 lakh molecular weight and are composed of 6 subunits. All these proteins are highly aromatic, containing a high proportion of tyrosine and phenylalanine residues. They are synthesized predominantly in the fat body, and their concentration increases enormously in later larval instars. They did not occur in the adult stage. Then heat shock proteins. Heat shock proteins are present in some of the insects, particularly Drosophila melanogaster. These are a specific group of proteins, and the synthesis of which is induced by heat and some chemicals of environmental stimuli.